Welcome to the fascinating world of molecular biology, a field dedicated to understanding life at its most fundamental level. This discipline is defined as the study of the molecular basis of all biological activity within and between cells. It delves into the intricate mechanisms that govern life. Our focus in molecular biology is on the tiny yet powerful molecules that orchestrate every living process. We explore the roles and interactions of DNA, RNA, and proteins, which are the primary actors in the cellular drama. These molecules hold the secrets to life itself. The scope of molecular biology is vast, exploring precisely how these molecules interact with each other and how they function within the complex environment of a cell. It seeks to unravel the precise pathways and feedback loops that maintain cellular homeostasis. Understanding these interactions is key to unlocking biological mysteries. The significance of molecular biology cannot be overstated, as it reveals how life's processes are meticulously orchestrated. From the simplest bacterial cell to the most complex human organism, molecular mechanisms dictate growth, development, and reproduction. It provides the foundational knowledge for all biological sciences. This field is incredibly important for comprehending cellular function in its entirety. By understanding how genes are expressed, how proteins are made, and how cells communicate, we gain insight into the very essence of life. It explains the machinery that drives every living cell. Furthermore, molecular biology is crucial for understanding disease mechanisms. Many illnesses, from genetic disorders to infectious diseases, stem from disruptions at the molecular level. Unraveling these molecular defects is the first step towards developing effective treatments and cures. At the heart of molecular biology lies a fundamental concept known as the central dogma. This principle, first articulated by Francis Crick, states that genetic information flows from DNA to RNA, and then from RNA to protein. It describes the basic flow of genetic information in a cell. DNA, or deoxyribonucleic acid, is often referred to as the blueprint of life. It contains all the instructions necessary for an organism to develop, survive, and reproduce. This remarkable molecule is the ultimate repository of genetic information. The primary function of DNA is to store this genetic information in a stable and inheritable form. It ensures that genetic traits are passed accurately from one generation to the next. DNA is the master archive of biological instructions. The structure of DNA is a double helix, composed of a sequence of nucleotide bases. These four bases are adenine, guanine, cytosine, and thymine, often abbreviated as A, G, C, and T. Their specific pairing rules form the rungs of the DNA ladder. RNA, or ribonucleic acid, is a similar molecule to DNA but with key distinctions. It plays several vital roles in gene expression and regulation. RNA acts as an intermediary and a functional molecule. A crucial distinction of RNA is that uracil replaces thymine as one of its four nucleotide bases. So, RNA contains adenine, guanine, cytosine, and uracil, or A, G, C, and U. This difference is important for its various functions. RNA primarily serves as a messenger, carrying genetic instructions from the DNA in the nucleus to the ribosomes in the cytoplasm. This messenger RNA, or mRNA, is a temporary copy of a gene. It ensures that the genetic code is accessible for protein synthesis. Once created, this RNA carries the genetic code to the ribosomes, which are the cell's protein synthesis machinery. Here, the information encoded in the mRNA is translated into a sequence of amino acids. This process is fundamental to all life. Transcription is the initial step in gene expression, where the information encoded in a segment of DNA is copied into RNA. This process is carried out by an enzyme called RNA polymerase. It ensures that the genetic message is accurately transferred. Translation is the subsequent process where the genetic code carried by messenger RNA is decoded to produce a specific protein. This complex molecular event occurs on ribosomes. It is the ultimate expression of genetic information. During translation, the mRNA is read in sequential 3-base sequences, which are known as codons. Each codon specifies a particular amino acid or signals the termination of protein synthesis. This triplet code is universal across most life forms. Each codon specifically dictates which amino acid should be added to the growing protein chain. The precise order of these codons determines the unique sequence of amino acids in a protein. This specificity is vital for protein function. Transfer RNA, or tRNA, plays a crucial role by carrying specific amino acids to the ribosome. Each tRNA molecule has an anticodon that is complementary to an mRNA codon. This ensures the correct amino acid is delivered. The tRNA molecules recognize and bind to their corresponding codons on the mRNA template. This precise pairing ensures that amino acids are assembled in the correct order. It is a critical step for accurate protein construction. As tRNAs deliver amino acids, the ribosome links them together through peptide bonds, forming a long polypeptide chain. This chain is the nascent protein. The process continues until a stop codon is reached. Finally, this newly formed polypeptide chain folds into a specific three-dimensional structure, becoming a functional protein. The protein's unique shape determines its biological activity. This folding is essential for its proper role in the cell. DNA replication is a fundamental process that ensures the accurate duplication of genetic material. This is essential before a cell divides, so each daughter cell receives a complete set of chromosomes. It guarantees genetic continuity. This vital process occurs during the S phase of the cell cycle, preceding cell division. It ensures that every new cell has an identical copy of the organism's genome. Without accurate replication, genetic information would be lost. A key enzyme in DNA replication is DNA polymerase, which adds new nucleotides to the growing DNA strand. It reads the template strand and synthesizes a complementary new strand. DNA polymerase also has proofreading capabilities to minimize errors. Several supporting proteins work in concert with DNA polymerase. These proteins help to unwind the DNA double helix and stabilize the separated strands. They ensure the DNA remains accessible for replication. Errors during DNA replication can have significant consequences, leading to mutations. These changes in the DNA sequence can alter gene function, potentially causing disease or affecting an organism's traits. Accurate replication is therefore paramount. Gene regulation refers to the intricate control of gene expression, determining which genes are turned on or off, and when. This allows cells to respond dynamically to their environment and differentiate into specialized types. It is a highly sophisticated system. Cells possess sophisticated mechanisms to turn genes on and off in response to various internal and external signals. This precise control ensures that only necessary proteins are produced at the right time and in the right amounts. It conserves cellular resources. Regulatory elements, such as promoters and enhancers, play a critical role in controlling the binding of transcription factors. These factors are proteins that either promote or inhibit the initiation of transcription. They act as molecular switches for gene activity. Molecular biology has profoundly revolutionized numerous fields, including medicine, agriculture, and biotechnology. Its discoveries have transformed our understanding of life and our ability to manipulate biological systems. It continues to drive innovation across these sectors. One key technique that exemplifies this impact is the polymerase chain reaction, or PCR. PCR allows scientists to amplify specific DNA sequences exponentially from even tiny samples. It is an indispensable tool in modern biology. PCR has a wide range of applications, from diagnosing infectious diseases to forensic analysis in criminal investigations. It enables rapid and sensitive detection of genetic material. This technique has become a cornerstone of molecular diagnostics. The principles of molecular biology form the fundamental basis of genetic engineering. This powerful technology allows scientists to directly manipulate an organism's genes. It has opened new avenues for scientific research and practical applications. The primary purpose of genetic engineering is to modify organisms for desired traits. This can involve introducing new genes, altering existing ones, or removing specific genetic sequences. It offers unprecedented control over biological characteristics. The outcomes of genetic engineering are diverse and impactful, including increased crop yields in agriculture and the development of new therapies for human diseases. It has led to the creation of genetically modified organisms with enhanced properties. These advancements hold immense promise for the future. Overall, molecular biology provides a fundamental understanding of life's processes at the most basic level. It explains how genetic information is stored, expressed, and inherited. This knowledge is essential for all biological inquiry. Looking ahead, molecular biology continues to pave the way for groundbreaking discoveries and innovations. From personalized medicine to sustainable agriculture, its ongoing research promises to address some of humanity's greatest challenges. The future of biology is deeply rooted in molecular understanding.